Like I said, if you're watching the videos as opposed to joining live stream or being here in person, sometimes you have to jump through a few extra hoops. And in this case, your few extra hoops is watching a minute or a video that's about 5 minutes longer than the normal lecture. And on that note, the first word for attendance, not that any of you guys have to have it unless you're watching the video, will be early. Early is the first word for attendance. So yeah, if you guys send in early, you'll get a little bit of extra credit. And for those of you who are online right now, you will also finally get a little bit of extra credit, which isn't normally something that we have for online viewers, I guess you could say. So yeah, early is the first word. Anyway, let's get to this here. Let's see. Oh, Kincaid. Andrea. Mullins. Lewis. Cooper. Hunter. Both Meltons are here. Crowder is here. Reinhardt. Again, both Meltons are here.
Morning.
Morning. Julia is here. Rogers. Talbert. Kennedy is here. If you're wearing WVSU gear, and I don't say it when I say your name, point it out to me. It needs to be obvious, like mine or hers. So if you're wearing WVSU socks or underwear, just keep it to yourself. Kylie's not here yet. Larissa. Burford? Good. I don't know why I have such a. I think it's because I lived in Beaufort for so long. So every time I read your last name, my mouth is wanting to say Beaufort. Anyway, she's here. Madeline. Hughes is here in WVSU gear. Galloway. Cook is here. Durgan is online. So to remind you guys, I don't think I've had any problems with you guys online, but just as a reminder, if you're online streaming right now, don't forget you have to send the words before 9 a.m. I know there was one person who seems to be like they attend online but don't send the words. So remember, you have to send the words because it's very easy to log in online and then go back to sleep. Anyway, Tate, not yet. Burton is online. Kinder. Jones. Canterbury is here. Kraus. Hobson. Ray. Fisher is online. Got 2 minutes and 1 minute, some change. We go back to the top here. Abigail is here. Kincaid is online. Andrea is not here yet. Mullins is not here yet. Lewis is not here yet. Cooper is online. Hunter is here. Both Meltons are here. Crowder is here. Reinhardt is here. Julia is here. Rogers is not here. Talbert, not here. Kennedy is here. Hannah is not here yet. Larissa's not here yet. Madeline's here. Madison is here. Galloway is not here. Cook is here. Durgan is online. Tate is not here yet. Burton is online. Kinder is not here yet. Jones, not here yet. Canterbury is here. Prowse, not here. Hobson, not here. Ray, not here. Fisher is online. Larissa came in and is physically here. And then, let's see, Andrea, right? Okay. Anybody I didn't call? Everybody seems to be accounted for. Well, yeah, at least for attendance. Some people are still missing. All right. So we'll talk about lab really quick because we're having some issues. And I can see why, because you guys are all getting used to it. It is complicated. I try for it, like I'm torn between trying to make it complicated but giving you a bunch of options. And I know it's complicated because you got to open this and make yourself a copy and share it. So I am being really loose, right, as far as some of these requirements. But we're going to start cracking down. So let me explain some of the stuff. Some of this is a refresher and some of this is new information, right? And I apologize for the changes. Hopefully this will help streamline things and make it to where I can grade things quicker, which will help you and me. But anyway, the first thing is be on time. I've mentioned this a lot. It's okay. That's not okay. You shouldn't come in late anywhere, here or lab, but especially lab. Lab's important. Like we're starting this. We're all in it as a team. So if you come in late, not only are you distracting me and your classmates, but you have no idea what's going on. And then we're going to be working as a team, and you're going to be the slow person, slowing everybody down, if not screwing things up. So be on time. Another thing, if you're going to be late, you should let me know. Because remember how many you guys notice I take attendance, right, at lab, because if you're not there, you don't get credit. I take attendance, then we start, and then I guess a couple of people have slid on into the lab without me noticing and haven't told me. So they have a 0 for their lab, or at least partial credit, because they weren't there. According to my records, they weren't there. So if you do come in late to lab, make sure you let me know. It's so important. It's also important to let me know if you're coming late to here, but here it's like 2.4 points. And if you were really that worried about it and you came in late and forgot to tell me, you could just watch the video and get your points that way, right? So here it's a little bit less important. Very important on lab. Pre-labs. Make sure you do the pre-labs. That's really, really important. The idea is for you to understand what's happening before you come to lab. Because if you don't, same thing I just said earlier. If you're lost, then everyone else is going to be, you're going to be behind. You're going to be affecting your teammates. So make sure you're there. And look, there's pre-lab videos. I don't know how to make them more obvious. Like if you go to the pre-lab for this week, it says pre-lab. Make sure you watch this video before you do these answers. So I'm doing my best to make them obvious. So look really carefully when you do the pre-labs and make sure you do them beforehand. Which brings me to due dates. Again, we're kind of easing into it. So this is the 4th week we're coming into next week. So I think now it's time to start enforcing due dates. Get your pre-labs done before, by Sunday. It's not going to be deduction of points, but if you don't have your pre-labs from now on, you can't come to lab. And if you can't come to lab, you get a 0 for the lab, right? So get your pre-labs done on time. Let me know if you need help. Those due dates are very important. Matter of fact, I would prioritize pre-lab due dates before lab due dates. For example, all lab 3, right, the one we're doing this week, it's due Friday. Lab 4's pre-lab is due Sunday. But if I were you, make sure you get that pre-lab done. Because I'm not going to be so strict on the lab due dates as far as like the ones that are due on Friday. That makes sense? I'm going to be a little bit looser on those. That's important. It's really important that you know what's going on before you come to lab. So pre-labs, I'm going to be pretty strict with for that reason. Attention to detail, that kind of goes back to this stuff. Everything I've said so far, right? So we already know about the due dates. So if you had attention to detail, you would know about the due dates. The videos, right? Make sure you're paying close attention. Yeah, that's it for that. Sharing. So one of your classmates left some great, at the end you have extra credit where you can offer suggestions, and one of your classmates offered a great suggestion. So you guys can work together. Yes, in lab, sometimes it'll be just me doing a demonstration. Sometimes you'll break out into teams. Sometimes you'll work individually. Whatever the case may be, once I'm done with that, you guys are all welcome to mingle about. Work with each other. Ask questions like, hey, what'd you get for this? Does this look right? Don't copy, but you can still confirm, right? Say, look, compare what you got to what they got, and, see who's correct, so on and so forth. So, yes, not only can you work and share together, I recommend it. Everybody good there? Actually, that's not what I meant for that bullet point. Just jogged my memory. So another thing that's a pain for you guys, and this isn't a big deal because if you fail to do it, I'll do it. But if you can remember, when you share those labs, share them with me and also your TA now. So that's a new thing. Share it with your TA. If you can't remember, I'll fix it. Don't worry about it. I'm asking you guys a lot, so if you forget that, whatever. Corrections, again, to streamline, to make things quicker, because I'm behind on grading and that's not fair to you. From now on, I'm not going to ask you or remind you that you can make corrections. So this will be the last time. Just know it, right? You can always make corrections. So instead of me reminding you, like, hey, if you make corrections, click on this and let me know you made corrections so I can regrade it. Just go ahead and do it, and go ahead and click on it and let me know. Does that make sense? I don't even need to remind you more. It's a given rule for the rest of the semester. You can always turn in things, make corrections, including if you just didn't do it, right? If it's late and you just didn't do it, you can still do it. Do it, turn it in late, leave a comment, or reply to one of my comments that says, hey, I'm ready for you to grade this. Is that good? All right. And then finally, what was the other one? Feedback. What was that? Why did I even write that? Oh yeah, same thing, right? So again, because I'm behind on grading, which isn't fair to you, I'm going to try to streamline the feedback. So just to remind you guys, because I'm going to be a little bit abridged on my feedback, you can always ask me. So I'll give you a grade, and if you want, like, I do want to fix that, but what's wrong with it? Then ask me. You know, like if I marked -1 on something, feel free to click on that and reply, what's wrong? And I'll let you know. That make sense? But some people don't care, and I don't blame them. You got a lot of stuff going on. You guys aren't biology majors. You have more important classes. So I don't want to leave like long feedback saying how you can fix it to somebody who's just going to be like, oh, whatever, that's a pretty good grade, I'm going to leave it. That make sense? I'll save my time, I'll save your time. If you want feedback, let me know. That's it for lab. Again, I'll remind you, be on time for lab. We'll be on time for everything. It is distracting, but lab is even more important. You can't be late to lab from now on. And if you are, by goodness, like I said, make sure you let me know. So any questions about lab? Such a pain, I know. We'll get it. So while I'm pulling everything up, let's talk about the plan of the day. We are going to finish chapter 2. Chapter 2. In chapter 2, what's left is not much. So it should be pretty quick. Which means we will be done with the material that's going to be on the exam today. Unless something goes horribly wrong, like there's an explosion or a fire or a chemical leak, we have to leave early. But yeah, we'll be done with that. So obviously I'm not going to say spring it on you and tell you to have an exam on Friday. So start thinking now. The exam will definitely be sometime next week. Sometime between now and then, I'm going to send you a message and ask your input and let you vote on which day you want it to be. I'm still fighting to get it to be online to where it doesn't have to be a certain day of the week where you can just log in and take it whenever you have 50 minutes to take it. So I'm fighting for that. But for now, let's just plan on it being one of the days, Monday, Wednesday, or Friday next week, and you get to choose which one. Good on that? All right. Uh-oh. I think I looked at this thing the wrong way, and it turned off. Oh, we're good. All right, let's get started. Back into it. And obviously, I was really, really tired and a little sick last time we met. So anybody needs any clarification from any of the material that I tried to go over last time we met, just let me know. I'd also remind you as I'm pulling this up, study guides, do your study guides. You don't have to, but I use the study guides to make the exams. So if you do the study guide, you'll have seen almost every question on the exam, some version of it, because I will reword them. But anyway, here we go. So to remind you, again, the big picture, this portion of the chapter, we're talking about biological molecules. And the big four categories we've talked about so far are carbohydrates, and we talked about lipids, and we talked about proteins, and then finally we're on the last category talking about nucleic acids. And really quickly, I'll back up and show you what we've talked about so far. That was proteins. Yeah, this is it. This is all we've done, right? So we've only done the introduction. We already talked about what nucleic acids do, which would be more important later in the semester, less important for this exam. And then the two types, DNA versus RNA. And again, we'll go into more detail about that later in the semester. The first word for attendance, the first regular word for attendance, I should say, will be, let's say, proteins. Anyway, a gene, I think we already talked about this. I know this is where the last slide we talked about, but anyway, really quickly, a gene is a unit of inheritance this coded in a specific stretch of DNA. Yeah, we did talk about this. Yeah, like I said, I summarized it by saying a gene, the way you should think of a gene is its instructions on how to build a protein. So we already know that we're talking about nucleic acids. We're talking about DNA and RNA, and that's where you find genes. And genes are things that provide instructions on how to build proteins. However, for this exam, I'm not going to ask you what a gene is. There will be one whole chapter when we talk about how genes work and the structure and function of DNA and the structure and function of RNA. So for now, just consider that an introduction. We'll talk about it in greater detail later. Again, this is another picture trying to show you, if you were to look at a big long strand of DNA, which that's a short strand, imagine that thing was a lot longer, and then we zoomed into this one part, and then we take that gene and we turn that into a gene of RNA, and then we take that gene of RNA and we turn it into a protein. And again, this is all just an introduction because we will be covering that in a later chapter. Nucleic acids are the key macromolecule in the continuity of life, because that's all the information, right? DNA, in a sense, is what makes you, at least physiologically, right? Because you are built, your body built itself. I mean, it was started in the womb, but the instructions on how to build you were inside of you, and then you built you, right? So that's what we mean by it's the key macromolecule in continuity of life, because again, it carries genetic info. Again, those genes that provide instructions on how to build you. And there's two main types, like we already said, DNA and RNA. This is a great time to remind you, you don't need to know the full name yet. Maybe when we talk about it later in the semester, we'll talk about deoxyribonucleic acid and ribonucleic acid. But for this chapter, DNA and RNA, the abbreviations are just fine. And all this other stuff is not really important. It's important. It tells you why we're talking about it. But as far as this exam is concerned, that's not important information. This is stuff we're really going to dig into later. So any questions so far? Might have an early day. Regarding DNA, yeah, this probably won't be on the exam either. Not this one, but the later one. But here it is anyway. DNA is the genetic material, and it's found in all living organisms. And RNA is used mostly for protein synthesis. And we're going to talk a lot about that later in the semester. But for independent work, if you want to, you can start looking it up now, which is this. Like I said, the RNA is mostly used for protein synthesis. And there's a few other exceptions that we'll talk about. But then there's a lot of other exceptions we don't talk about. So if you want to, for independent work, you can look up the other things RNA does. Of course, if I were you, I'd wait until later in the semester when we're actually talking about it, but it's up to you. And also, DNA also uses RNA to communicate with the cell. That's all stuff we're going to cover later in the semester. So again, this is just an introduction. This is how your textbook authors wrote it. They provided all this information at the beginning and then repeat it later, which I think is dumb, but whatever. All right, here we go. Back to the stuff that you need to know, right? This theme of all the other stuff. We're talking about what's the polymer, what's the monomer. The polymer is called a polynucleotide. That is the polymer. So again, to remind you, the first thing we talked about carbohydrates, and the polymer for that was polysaccharides. Then we talked about lipids, which just don't technically have polymers and monomers. Then we talked about proteins. The proteins polymer was polypeptide, and the monomer was the amino acid. And finally, the last category, nucleic acids. The polymer is the polynucleotide, and the monomer is the nucleotide. It's nice and easy. I think nucleic acids and carbohydrates are the easiest because it's in the name, right? For carbohydrates, you have monosaccharide and polysaccharide. It's in the name. For nucleic acids, you have nucleotides and polynucleotides. Yeah, so don't miss that. Someone's going to miss it. But remember, the only one that has a weird polynucleotide name would be proteins, right? Amino acids are the monomers, and you put a bunch of amino acids together, and you get a polypeptide, right? It's confusing. If it was up to me, I would change it to be like amino acids and polyamino acids, but I'm not in charge. So anyway, does anybody have any questions about those two bullet points? You need to know the monomer and the polymer. All right, so if you remember when we talked about proteins, we're like, all right, the monomer of a protein is amino acid, but then we didn't leave it there, right? Then we talked about the parts of the amino acid. You have to know the different parts of the amino acid and which part makes it different than the other 20 amino acids. Well, we have the same situation here. So let's talk about the 3 parts of a nucleotide. Whether we're talking about RNA nucleotides or DNA nucleotides, they have these parts. A 5-carbon sugar, a phosphate group, and a nitrogen-containing base. So you need to know those 3 parts: sugar, phosphate, and base. There might be some questions about this on this exam. There will definitely be questions on this when we cover it in more detail later in the semester. So yes, and I bolded these words because I like to, if you're doing flashcards, this will suffice. 3 parts of a polymer, 3 parts of a nucleotide: sugar, phosphate, base. If you remember that, you're fine. Because on the multiple choice exam, I'm not going to say, what kind of sugar is it? Is it a 5-carbon sugar or a 6-carbon sugar or a 4-carbon sugar? I'm not going to just know that it's a sugar, phosphate, and a base, right? So I'm not going to get specific and try to trick you with the number of carbons for the sugar, or the fact that it's a phosphate group and not just a phosphate, or the fact that it's a nitrogen-containing base and not a, silicone-containing base, or whatever. So sugar, phosphate, base. Memorize those 3 words. Any questions about that? All right, so if you remember from the amino acids, remember there was 4 parts to an amino acid, and I said there's only one part of that amino acid that kind of defines it. It sets it apart from the other 19 amino acids, because remember there's 20 amino acids. So here, the only part that varies, the part that defines what kind of nucleotide you're dealing with, is the base. So the base is the part that varies. That's the variable part, and the base is the thing that defines it. So no matter what, if you're looking at DNA, all the nucleotides, whichever kind you're looking at, has the same kind of sugar. All the nucleotides, they have the same kind of phosphate group. But each individual nucleotide has a different base, because that base defines what it is. Any questions so far? I'm going to point to the next attendance word, draw an arrow there. I'm not going to say it. So if you happen to be watching this online, relaxed with your eyes closed and just taking it all in, which is completely fine. Open your eyes, look at the screen, look where the arrow is pointing. That's the next word for attendance. All right, here's some more pictures. These aren't from your textbook. So here we're looking at a nucleotide. There's 2 different ways of looking at it. We're looking at the same thing, just drawn different ways. We've seen that a lot in this class, right? So there's the phosphate group, there's the sugar, there's the base. Right? That one gives it a better idea of like what's bonded to what. This is a more simplified version, but it's the same thing, right? Phosphate, sugar, base. Even the base is the same. That's a T, this is a T. Those are different ways of drawing the same thing. I'm definitely not going to show you a picture and say which one of these is the sugar, which one of these is the base, which one of these is the phosphate group. I'm not going to ask you that. But there it is. Any questions? All right. So ACGAGTC. Okay. If we're talking about DNA, it only has, like I said, or let me back up a step before I say that. So to remind you, if we're talking about amino acids, which is a protein thing, there are 20 different amino acids. If we're talking about DNA, there's only 4 different types of nucleotides. Same thing with RNA, but they have different types. So we'll talk about one at a time. The 4 different types of nucleotides for DNA are adenine, guanine, thymine, and cytosine. That's not in any particular order. It's not like one's better than the other or more abundant than the other. That's just the order in which they were written. So don't read into that. Another thing for the exam, especially for the first exam, maybe even for the second one, is I'm not going to require you to know the full name. For now, just the letters will do. A, G, T, and C. And I'm not even sure I'm going to ask you about that yet, because again, we're going to have a whole chapter on this. So I don't want to, this is my lowest priority when I'm writing your exam. I'm going to have more questions about other stuff than the specifics about nucleic acids, because we're going to learn about it later. But are there any questions about this slide? All right, I'll show you a picture of the A, G's, and C's and T's. That's what they look like. Notice that these are double ring and those are single ring. That will be important later, but for now, it's just an introduction. And then, of course, here's a strand of DNA, and they've kind of pointed out the different bases, the C's and T's and G's and A. But again, even in the next exam, I'm not going to show you a picture of these and be like, which one of these is adenine? Which one of these is cytosine? So this is just for your own knowledge. Any questions about this slide? Okay. As you might imagine, it's a dehydration reaction that brings these nucleotides together. So there's nothing new there, because like I said when I first told you what a dehydration reaction is, as far as we're concerned in this class, anytime we bring two molecules together in a chemical reaction to make a bigger molecule, we're calling that a dehydration reaction. That's what changes the nucleotides to polynucleotides, right? So here's a nucleotide, here's a nucleotide. I'm going to put them together in a dehydration reaction, so now they're covalently bonded, and that is now a polynucleotide because you have more than one nucleotide. This is another one that'll be important later, but not for this exam, so I'll put it next to it. Like I just said, it's a covalent bond, which is really important for later, but not necessarily now. And also, like I said, there's three parts, right? There's a sugar, a phosphate, and a base. So just so you know, those covalent bonds in these dehydration reactions, that's made between the sugar and the phosphate. So the base is just sitting there flapping around, so to speak. It has a purpose. We'll talk about it later. But it's the sugar and the phosphate that are bonded to each other. So a sugar is bonded to a phosphate, which is bonded to another sugar, which is bonded to another phosphate, so on and so forth. But again, that'll be more important than later when we really talk about this in more detail. And then you'll definitely need to know this bullet point, sugar phosphate backbone. But for now, it's not very important. But again, your textbook included it, and this one didn't. Sorry. Anyway, any questions about that? That will be important later. All right, there are some differences between DNA and RNA, so let's talk about some of them. First of all, DNA is double-stranded. You need to know that. Again, if you were writing flashcards, that would be enough. DNA on one side, double-stranded on the other. But to explain what I mean by double-stranded, not that I'm going to ask you what double-stranded means because it's pretty obvious, but here it is anyway. It means those polynucleotides are coiled around each other, and it forms something called a double helix. And again, we'll talk about that later in the semester. Of one strand of DNA are hydrogen bonded to the bases of the other strand, which is another way of saying one strand of DNA is hydrogen bonded to the other strand. And then, like I said in this first bullet point, then they kind of coil around each other. That's where you get the twist. And that will be very important later, but not for this exam. Same thing with this. I'm almost definitely not going to ask you this on this exam. You'll definitely need to be very familiar with it on the later exam. But the bases always pair up with each other. Because like I just said here, right? One strand of DNA is hydrogen bonded to the other, and they're bonded by the base, right? One base hydrogen bonded to the other. And they have rules. A's only pair with T and vice versa, and G's only pair with C and vice versa. So later you would definitely need to know that A's go with T and G's go with C. But for now, just an introduction. Any questions about that slide before I show you a picture of what's on that slide? All right, there we go. So again, when you're talking about a strand of DNA, or excuse me, a molecule of DNA, what you really have is two individual strands of polynucleotides that have come together in hydrogen bonds, and then it's twisted. So a DNA double helix is made up of two strands of DNA. And again, that'll be more important later in the semester. Any questions so far? Moving right along. We might have a good early day. Sorry, just as a reminder, if you came in late, as always, make sure to remind me. Send me an email and tell me you came in late, because I forget if you came in before or after I made that reminder. Anyway, again, differences and likenesses are similarities between DNA and RNA. And these are questions that you'll probably get on both exams. Again, whether they're the same, both DNA and RNA are polymers of nucleotides. I've already told you that. That's just a refresher. That's something they have in common. All three of them have, both of them have a sugar and a phosphate and a base. Now, the sugar and the base is a little bit different, but we'll talk about that later. For now, just know that these are how DNA and RNA are the same. Right? They're both nucleotides, or both polymers of nucleotides, and they both have a three-part nucleotide that consists of a sugar, phosphate, and base. Right? 5-carbon sugar, phosphate group, and a nitrogen-containing base. And you can see it on the pictures here, right? There's the basis for RNA. There's the basis for DNA. There's the strand. There are two strands of DNA. There's one strand of RNA. Any questions about that slide? All right, let's talk about the differences between DNA and RNA. First of all, the sugar is different, hence the different name. So for RNA, like I've said before, that's called ribonucleic acid because the name of the sugar is ribose. Unlike DNA, it has deoxyribose. That is definitely a detail. I should say definitely. Almost definitely a detail I'm not going to ask on the first exam. They do have different bases. So like I said earlier, I told you the bases for DNA are A, C, T, and G. But for RNA, there is no T. There's only a U instead. So there's a new one called uracil. Well, new to this discussion. And of course, like you already saw in that last picture, RNA is single-stranded, DNA is double-stranded. So know those differences for the exam. Are there any questions about that? All right. Here's another picture. Again, so we're talking about this one's RNA because the way I can tell is, first of all, this sugar is a ribose instead of a deoxyribose. So that's one indication. And the other thing is this is a U. Right? The base is a U. DNA doesn't have a U. It has a T instead. So again, I'm not going to show you a picture and ask you to identify the parts. But if I did show you this picture, I could ask you, is this RNA or DNA? Because again, those two things should stick out to you. It has a different sugar and it has a different base. Right? That ribose sugar and that uracil base, that makes it RNA instead of DNA. Any questions? Yes.
Like the T, the thymine, I think that's how you pronounce it. There is like a carbon tree and like a oxygen. That's really the only difference with the uracil I noticed. How come that makes the big difference between RNA and DNA?
Just because that's the only base that they have different. So whether it be like what you just described or anything, even if this carbon was single bonded to that oxygen instead, and then there was another hydrogen, that would be a whole different base. It's not a T, C, or G, or U. But whatever it is, if it's different, that's just what makes it different, right? So because it has that different chemical structure, that's what makes it a U instead of a T, and therefore that's what makes it RNA instead of the DNA. And it's not specifically that, in that case, that is specifically what makes it different, but anything can make this different. Any slight change to this molecule or any of the other ones we discussed, and it's no longer that molecule. So like those molecules that you drew for lab last week, if you messed it up, technically you drew a different molecule. But anyway, good question. And if you want, we can dive deeper into that later or look into it for independent work. But yes. Any other questions? That was a good one. All right. So anyway, if you download the PowerPoint, when you get to the end of it, there's this big link right here. You can click it for the Chapter 2 study guide. Let me see. And I'm going to, if I can do this quickly, at least bring up the next chapter, do the introduction, and then call it an early day. See if I'm still logged in here. Hey, I am. That'll work. Chapter 1, Chapter 2. Any questions while we wait? Actually, that'll be the next attendance word. Wait, because we are waiting. Oh, while we're happy here, why not do this? We're recording. You're looking. So, of course, this is a PowerPoint, and this is not a lab or an independent work, but it's still going to work the same. What you're going to do when you open up the document, you go to the top right. Well, first of all, you make yourself a copy. So file, make a copy, make yourself a copy. Then you share it with me, because I'm already on there, and then Elijah. And if you he's at the top of my list anyway, but if you start typing Elijah, his name will pop up. But anyway, there we go. It's been downloaded. Let's open it up very quickly. So the good news is, like I said, where'd you go? I just downloaded you. There we go. So no matter what, we're not having the exam on Friday, which is great. That means even though on Friday we're going to start new material, which is this slide, this PowerPoint, but it won't be on this first exam. That means you have a full weekend to study in addition to the rest of this week. So hopefully you guys will be really prepared next week. And like I said, I'm hoping to get this thing working to where you can just go online anytime next week, anytime you have 50 minutes, and just take it on your own. Otherwise, we're going to pick a date to come in and do it together. You need to bring in your computers. And it is timed. So if any of you want more than 50 minutes to take it, whether it's in person or there, then you need to contact me sometime between now and then and say, let's find a time for us to meet online where you can share your screen and you have your microphone on and have your camera on. If I could see you and make sure you're taking a closed book exam and you're not looking things up or using your book, I don't care if it takes you 5 hours to take an exam, right? If we're there and I can see you and I know it's a closed book exam, I don't care how long it takes you. So keep that in mind. If you're like, oh, I get test anxiety and I don't like being rushed, you have options. Contact me. We'll make arrangements. Anyway, really quick. Chapter 3, structure and function. Cell structure and function. This is the next portion, right? So now we're done talking about the small stuff, the molecules and all that. And now we're going to talk about how cells actually work. And then finally, after that, we'll get into the better stuff, in my opinion, which is how organisms interact with each other. But the next portion will be about cells. And here's how it's going to be broken down. First, we're going to talk about how cells are studied. Then we're talking about comparing prokaryotic and eukaryotic cells. Then we'll talk about eukaryotic cells specifically. Then we'll talk about the cell membrane, passive transport, and active transport. And I'll go ahead and tell you now, in my opinion, I would say this is the most important part as far as exams are concerned. Because all this other stuff, yeah, like this is a good introduction to what we're going to talk about. This information will be good because a little bit of it will be used throughout the semester. Same with this one. Actually, in this one, 3.3, and 3.3, we're really going to talk about our cells. Like our cells are eukaryotic cells, and we'll really talk about how they work. But in my opinion, it's not that important, which probably sounds weird, right? Because like I said, life is cellular. Cells are all about biology. But later, we're going to talk about cellular reproduction and cellular respiration and photosynthesis. So all the really important things that cells do, we're going to have a whole chapter on it. Therefore, I'm going to skim past this really quickly because why do I need to go into detail about all the little things that cells do when later we're really going to go into detail about all the important things they do. So, yeah, you know what? Let's do 3.1 because like I said, it is a good introduction and there's not much information that's going to be on exam 2. So let's just knock the introduction out of the way. And when we come back Friday, we can start with the meat and potatoes. The learning objectives of the first portion are to describe the roles of cells and organisms, to compare and contrast light microscopy and electron microscopy, which I'm teaching out of the textbook, which is the only reason I'm bringing that up because I'm not going to ask you any questions about that. You completely could brain dump everything I'm about to teach you in that slide, and you'll be completely fine for the rest of the semester. And then also summarize the cell theory. So here we go. Microscopy. There are 3 different types of microscopes that we're concerned with here. There's light microscopes, dissecting microscopes, and electron microscopes. If you're a note taker, no need to take those notes. I'm just teaching out of the textbook to keep things in order, I guess, because I'm not going to ask you any of these. I'm certainly not going to ask you all these parts. I'm working on making an online virtual lab for microscopes because I'm not going to do a microscope lab in this semester. So hopefully I'll have that up and running for you if you want to do it for extra credit. But anyway, those are the 3 different types of microscopes. As you can probably imagine, each one of them has a lot of categories. Like there's a lot of different types of light microscopes. Some of them have one thing to look through. Some of them have 2. So some of them use electricity. Some of them use mirrors. Well, they all use mirrors. But anyway, there's different types of those, but those are the 3 main categories. Cell theory, bless you. That is the unified cell theory, states that all living things are composed of 1 or more cells, and that the cell is the basic unit of life, and that all new cells arise from existing cells. You should know that for exam 2, right? So again, to remind you, everything we're talking about now will not be on the first exam. This is new material. With that being said, this is the only new thing I've told you. I already told you from chapter 1 when we talked about the properties of life, you know that all living things are composed of cells, 1 or more cells. And you know that the cell is the basic unit of life, so that's all a refresher. Only thing new here, and you probably already knew it, was that all new cells arrive from old cells, right? Cells just don't magically appear, though they did at one point That's a whole different discussion that we'll talk about when we talk about the origins of life and about evolution. But for now, this is just an introduction. We won't get into that yet. Leeuwenhoek was the first to observe bacteria and protozoa. I'm certainly not going to ask you anything about him, but there are some videos out there I've shared, and they're interesting. Their stories are interesting. You can watch them and answer the questions for extra credit. Same thing with Hooke, right? Hooke, he's also known in the microscopy world. He coined the term cell because he looked at a cell. I think he was one of the first to see a cell, and it looked to him like a little prison cell. So that's why he called it a cell. Not that you need to know that. But anyway, both of them are credited to be the first two scientists to discover microorganisms. Not that I'm going to ask you. This is not a history of science class. Though there are some people where you're going to need to know the name, but they are not one of them. Anyway, if you're interested in them, you can look them up for extra credit. Or excuse me, independent work. Comparing prokaryotic and eukaryotic. Yeah, we can go through this really quickly. Because again, there's not going to be many questions, if any, on this. You do need to know the difference between prokaryotic and eukaryotic, mostly just because I'm going to use those terms throughout the semester. But most likely, I'm not going to ask you the differences. But here we go. Learning objectives of this section are to name examples of prokaryotic and eukaryotic organisms, compare and contrast prokaryotic cells and eukaryotic cells, and describe the relative sizes of different kinds of cells. So here's some things that they all have in common. Before we just start talking about the differences between prokaryotic and eukaryotic, let's talk about the similarities. All cells have plasma membranes. You definitely need to know what a plasma membrane is, again, for exam 2. It is the outer covering that separates the interior from its surrounding. I would say it's almost like a wall, but I can't use that term because there is actually something called a cell wall that we'll talk about later, and it's different than this. Just think of it like a oily blob, right? That's just what makes the cell, right? It's the envelope, the thing that keeps it inside, everything inside. It's filled with something called cytoplasm, which is the jelly-like region where all the other cellular components are found. And again, these are words that I'm probably not going to ask you about, but I'm going to use them, so I want you to know what they are. So that way when I use them, you know what I'm talking about. All cells have DNA, as we mentioned before. I think something interesting about the difference between prokaryotic and eukaryotic, which we're about to talk about later, is that we have different DNA. So for example, we have something called mitochondrial DNA. You have DNA in your mitochondria that only came from your mom, which only came from her mom, which only came from her mom. So if you look at that DNA, there is no paternal DNA involved in it, which I think is kind of interesting. We'll talk about that later too. All cells have ribosomes. And ribosomes are things that synthesize proteins. We'll talk about that. That is a whole chapter to discuss later. So for now, again, this is just an introduction. I would say there's not necessarily any reason to write that down, but I will use those terms, and the reason I'm bringing them up is because I want you to know what they mean when I use them. All right, let's talk about prokaryotic cells. Now that we know the similarities, let's talk about them separately. First one we're going to talk about is prokaryotic. There's basically two types, bacteria and archaea. And as far as we're concerned in this class, these two are almost identical. There are some differences. We'll discuss them. As far as I'm concerned, when you think of bacteria, you should just think of, prokaryotic cells. You should think of small, simple, basic, primitive cells. So there's a lot of domains, and that's a term you don't know yet, but I'll teach you soon. And 2 of the 3 domains are prokaryotic. But again, I'm not going to ask you that. That's an introduction to a later discussion. Again, like I said, when you think of prokaryotic cells, you should think simple and single cell, right? Very simple. They don't have a nucleus. That's one of the things you should know. Well, maybe. I don't know. Like I said, I'm not going to go deep into prokaryotic cells. Matter of fact, once we get past this chapter, we're almost never going to talk about prokaryotic cells again. They'll come up every once in a while. But everything we learn about is going to focus on eukaryotic cells. Right? When we talk about cellular reproduction and cellular respiration, we're going to be looking at eukaryotic cells, not prokaryotic. Anyway, in the prokaryotic cells, also the DNA is in the central part of a cell, which is a darkened region called the nucleoid. So even though I probably won't ask you this, that would be a great test question. Like which one has a nucleoid versus a nucleus? Right, so the prokaryotic cell has a nucleoid, eukaryotic cells has a nucleus. Prokaryotic cells are always, well, prokaryotic organisms are always unicellular. Eukaryotic cells could go either way. So any questions about this slide? All right. Now we're talking about eukaryotic cells, right? So now they have something called organelles, which prokaryotic cells do not have organelles. What are organelles? They are membrane-bound compartments or sacs with specialized functions. They're the things that do stuff in your cell. And again, I'm really going to brush past this because later in the other chapters, you're going to learn a lot about the very important organelles. So for this, I'm going to just rush through all the organelles and tell you that's what this one is, but you don't need to know it. This one does this, but you don't need to know it. So that part will be quick. The size difference is big. Like I already said earlier, you should know this. A eukaryotic cell is a lot bigger and more complex. A prokaryotic cell is a lot smaller and less complex. And here's like an example of looking at size differences. Like an atom, much, much smaller than these molecules, like a lipid and protein that you need to know for exam 1, which is, obviously smaller than the flu virus, so on and so forth. This is just something to look at. There's going to be no questions. There will be no test questions where I say, what's larger, a chicken egg or a frog egg? Of course, you could probably guess that anyway. But any questions about this slide? You certainly don't need to know these numbers. If anything, like I said, you do need to know eukaryotic cells are larger than prokaryotic, but I'm not going to get specific about how many micrometers are they. And that's where we'll end off. So when we come back on Friday, then we'll start getting into the more important stuff as far as exam 2 is concerned. So that's it for today. I'll see you in lab. Make sure if you haven't done the pre-lab, you do it, because you can't come to lab today unless you've done the pre-lab. And my goal between now and in lab is to be checking and rechecking pre-labs for you. And don't forget to email me if you came in late. Let's see.
Watch the 3 videos that you sent for the lab 2.
Awesome. Good. Lab 2, did you do the questions?
Yeah.
Good. Yeah, so make sure, well, I know you just told me, but go ahead and reply where it said reply when you did.
Okay.
Yeah, that way, when you do that, it sends me an email, and that's how I know to go in and open up the email and grade it.
Okay.
And then you sent me an email about my independent work, essay, and such.